Hi, my name is John Mascarenhas. I'm from the Icahn School of Medicine at Mount Sinai in New York, and today I'll be discussing the results of a safety analysis performed on the imetelstat studies, phase 2 trials that were conducted both in MDS, the EMERGE study, and myelofibrosis, the EMBARK study. So in these two trials, imetelstat, given as an intravenous infusion every 3 weeks at 9.4 milligrams per kilogram in myelofibrosis with 59 patient safety data, and in MDS patients at 7.5 milligram per kilogram every 4 weeks in 38 patients, was pooled for safety analysis. What we saw from this analysis that grade 3/4 thrombocytopenia in the myelofibrosis population occurred in 41% of patients, and grade 3/4 neutropenia in the myelofibrosis population occurred in 32% of patients. In MDS, grade 3/4 thrombocytopenia occurred in 61% of patients, and grade 3/4 neutropenia in 55% of patients. This led to less than 16% of patients having to discontinue therapy due to treatment-emergent cytopenias. And these cytopenias, I should mention, were short in duration, usually 1 to 2 weeks, and recovered counts prior to the next infusion. So these were manageable cytopenias. Now, grade 3/4 bleeding events occurred in 5 to 10% of patients across the studies, and grade 3/4 febrile neutropenia in less than 5%. So the majority of patients who had treatment-emergent cytopenias did not have significant clinical consequences as a result of this. There were grade 1-2 infusion reactions that occurred in 27% of the patients in the myelofibrosis study and approximately 13% of patients in the myelodysplastic syndrome study. So cytopenias were on-target based effects based on the mechanism of action of imetelstat. They were reversible and manageable. There was no evidence of imetelstat-related liver toxicity, and the grade 3/4 non-hematologic adverse events were basically unimpressive and rarely led to discontinuation. The phase 3 randomized studies, both in myelofibrosis and in MDS, will confirm the efficacy readouts of this novel telomerase inhibitor in myelofibrosis and myelodysplastic syndrome, as well as confirm the safety profile, which at this point looks very manageable with on-target cytopenias that rarely lead to discontinuation or a significant clinical consequence.